Assalamu alaikum. I'm Dr. Sheeba Akram. So let's start this important presentation on the human milk oligosaccharides. So we want to know what these human milk oligosaccharides really are. Today our main topic is as far as its impact on immunity is concerned. So we will talk about the immune system enhancement that's being caused by this. As we know that the infants and the newborns, they're quite deficient in immunity because their immune system is not properly well developed. So they are prone to any sorts of infections and diseases that may likely come to them because of their deficiency in the immune system development. So we definitely need something that will protect the newborns and the infants from different types of infectious diseases. So what we need to know and what we need for the infants. Uh, let's know what are the human milk oligosaccharides. Oligosaccharides, they are found in breast milk. It means they are provided by nature. Anything that is provided by nature is nothing less than the best. So these are the best. These are the best. Why? Because they are provided by nature. Nature never gives you anything that is less than the best. It is basically not a nutritious thing because it is not going to provide you with the calories, and it is not going to help you in growth and development in that way as some nutrient can. But it will play its role in immunity. How? Because it will stay in the gut. It will not be absorbed. 99% of the human milk oligosaccharides will stay in the gut, and they will not be absorbed by the gut mucosa. Only 1% of it will be absorbed. Rest of the 99% will stay in the gut. What it will do there by staying in that place, we will discuss it in the next slides. It provides specific advantages due to their complex structure. Structure has very important thing as far as its role is concerned. Over 200 types of HMOs has been defined by now, but the 2-fucosylated is the most common type that's been still we have recognized. Now comes the comparison of two things, and these two things are, one is HMOs, human milk oligosaccharide, and the second thing is prebiotics. Before giving the comparison, we must know what these prebiotics are. Prebiotics are basically the plant source fiber which acts as a diet for the good friendly bacteria. Good friendly bacteria means probiotics, which stay in the gut lumen of an infant, a newborn, or any individual, and they help us fight against the infectious diseases. So prebiotics are basically the food item for the bacteria, for the good bacteria, and they are from the plant source. While the second thing is human milk oligosaccharide, and they are also a source of help for the good bacteria, but they are coming from the bovine source, from the from the human source, from the mammal source. Now we compare the structures. As far as HMOs are concerned, they are highly different, completely different, very complex in their structure because they have so many branches, and they are much superior to the prebiotics. For example, what are the prebiotics? They are the fructo-oligosaccharides, which are obtained from the plant source, and the galacto-oligosaccharides, which are obtained from the cow's milk, the bovine source. These prebiotics, they can be very helpful nutrients for the probiotics, for the friendly bacteria, but not only for the friendly bacteria, but they can be equally good for the pathogen as well. So at the same time, they can give benefit to the friendly bacteria and for the pathogens. So in that sense, their efficacy and their value is to some extent gets compromised in that way. But as far as the prebiotics are concerned, their structure is very simple, linear structure, easily absorbed, and they can be a food item for the good bacteria and for the bad bacteria both. So in that sense, the HMOs, the human milk oligosaccharides, are much more superior to the prebiotics as far as this slide can prove. You can see it. Now we give a short review to the composition of the human milk. Human milk, as you can see in this picture, the protein is 8 to 12 grams per liter and the fat is 40 grams per liter. Lactose is, you can see here, 70 grams per liter and the oligosaccharides are 5 to 15 grams per liter. So it means that they are the third most abundant solid component in the human milk. What benefit it confers, we are going to see in the next slide. So next, let's see what benefits we are getting from these HMOs. There are 5 distinct benefits that we can get from these HMOs. First of all, it promotes healthy gut microbiota. As we all know that the human gut contains these microbiota which are very friendly. They protect you against the infections. And these HMOs, they promote that healthy gut microbiota. So this in that manner, it is indirectly helping us, but it has some direct antimicrobial effects as well, and it modulates the immune system, and it assists the gut barrier function, and it also prevents the adhesion of the pathogens to the mucosal cells of the gut. It doesn't let the pathogens to get adherent to the the epithelial cells of the alimentary tract, right? So that's how it help us in fighting against the infections. So let's see the promotion of healthy gut microbiota. Basically, these HMOs are the metabolic substrate for the beneficial bacteria. Anything which directly doesn't act, but if we give the example of an enzyme is inactive until and unless it has its attachment to some substrate, it gets attached with it. So in that sense, we can say these HMOs are the metabolic substrates for the beneficial bacteria. They provide beneficial bacteria the growth advantages, and they are structurally quite superior and distinct as compared to the prebiotics. In this slide, we can see that we are looking at the lumen of the intestinal tract of an infant, in which the epithelial cells are there, and the brush border can be visualized over the top of the epithelial cells, and we can see here 2 types of pathogens. The purple colored things, they are the pathogens, disease causing bacteria, and the green ones, they are the friendly bacteria which help you protect against the infections. When there are no HMOs, you can see that there is written over there minus HMOs, means there is no HMO over there. So both the pathogens and the friendly bacteria are present in the gut. But on the second picture, you can see that only green bacteria are present. It means when HMO is over there, it has removed the pathogens, and only the friendly bacteria, they are staying in the lumen of the infant's gut. Then comes the direct antimicrobial effects. Here you can see a picture of group B streptococcus. Group B streptococcus are basically gram-positive cocci. They are the bacteria. What they can do? They can cause neonatal sepsis. They can cause pneumonias. They can cause meningitis, which is a very severe sort of infection, and it is life-threatening. So these are very dire threats to the health of an infant, and these are very serious type of infections that can risk the life of a neonate, which are caused by group B streptococcus. And what these HMO can do over there, the HMOs, they have the ability to make the bacteria static. It has a bacteriostatic effect on certain non-sialylated HMOs. That's how it controls these infections. On the second slide, you can see Candida albicans. Basically, it's a fungal infection, and you can see the hyphae and the spores in the picture of the Candida albicans. Candida albicans has a potential to cause necrotizing enterocolitis. Majority of the doctors working in the pediatric department very well, they are aware of the fact that the necrotizing enterocolitis is a deadly disease of a neonate, of a newborn. What it does, it causes the necrosis of certain patches of the intestine of the infant newborn, and that's how it causes thereafter DIC can develop, and it can cause the death of the child. So it's basically a very dangerous thing for the baby, and it's caused by this fungal infection. And why it's there? Because of the lack of immunity. Immunity is direly needed over there to protect the infant against this Candida albicans infection. Otherwise, necrotizing enterocolitis can be fatal for the baby. Now comes the prevention of bacterial adhesion in the gut. Pathogens need to attach to the mucosal surface to cause infection, to cause infectious disease. This slide is very typical in the sense that whenever any bacteria has to cause a disease, it needs to be attached with the mucosal surface of the gut. There, after that, it is in a position to cause this disease. But the epithelial cell has a cell membrane which has a glycan element, and the bacteria gets attached with that cell membrane, and then it causes the disease. But when the HMO is not present, the pathogen has got all the potential to get attached with that glycan part of the epithelial cell and to cause the disease. We can see here where it is written that HMO is not over there, minus HMO. But when plus HMO means HMO is over there, oligosaccharides is over there. So oligosaccharide, by its presence in the gut lumen, is deceiving the pathogen, and the pathogen, instead of attaching itself to the glycan element, is attaching to the oligosaccharide. So pathogen with oligosaccharide, both of them get excreted and infection is prevented. Very simple and very logical mechanism of action. I hope you have well understand this. Assistance of gut barrier function. Most of the epithelial cells, they are not very tightly adherent to each other. So there is a space to move on. But these human milk oligosaccharides, they get embedded in between these gaps, and therefore they fill the barrier. They make the barrier complete for the pathogens to act. So the pathogens cannot penetrate in between these gaps. There is a direct modulation of the immune system. Basically, we are talking about the immune system. Immunity is the thing that is lacking. Immunity is the thing that is required. And we are advocating the human milk oligosaccharides just because of their very important and crucial role they are playing in the immune system for providing the immunity to the infants. Now, how these HMOs are helping in providing immunity to the newborn. They modulate the lymphocyte cytokine production. 1% of HMOs are absorbed into the systemic circulation. Potentially, they lead to a more balanced Th1/Th2 response, which is the immune maturation, And the HMOs, they stimulate the enterocyte maturation and immune reactivity, resulting in improved immune protection. So this is very obvious that how they are working by boosting the immune response. When the immunity of any infant is improved by taking the mother milk is the gold standard of nutrition for any infant. Nothing can be best than a mother's milk because nature has provided it with the best composition and with the best ingredients and with the best protectors. And uh, next to mother's milk, when any formula, because of any any sort of circumstances against which we are helpless, we cannot do anything but to provide the formula milk, we should go to the composition which is far close to the mother's milk, and it is only and only one milk formula that contains HMOs other than the mother milk. Mother milk definitely contain it, but there is just one milk formula other than the mother milk that also has been enriched with human milk oligosaccharides, and that is none other than the one NAN1. So next we will talk about NAN1, and for that I would request Mr. Zahid to come and say something about it. Thank you.